Okay, so last time we covered sympathetic nerves. So here we were trying to think about where our sympathetic nerves come from. Some of these new terms we have, preganglionic and postganglionic, and some of those pathways that the sympathetic nerves are going to take. So we're going to take some images and look at them in context with these questions. Okay. So first things first, our sympathetic nerves are going to come out of the thoracic and lumbar portions of the spinal cord. So the thoracolumbar region, so right in the center of your body. If we're thinking about where from within the thoracolumbar spinal cord they're coming out of a portion called the lateral horn, so out on the sides. And a preganglionic neuron, so number 2, is the first neuron to come out of the spinal cord. So it's the one going from the central nervous system out to the rest of the body. So that's the first point we would really consider this a peripheral nervous system. So our preganglionic neuron goes from the CNS out to a ganglion in the case of our sympathetic chain, or in the case of collateral ganglia, or in our special case for sympathetic nerves, out to the adrenal medulla, so the inside of our adrenal glands. So those are preganglionic neurons. We're sort of pairing those up with our presynaptic neurons in our head. This is a little different because we're going out to ganglia out in the body, whereas we saw presynaptic neurons, for example, in nerves that were entirely in the central nervous system, like in our brain. So this is just a little more specific, our preganglionic neuron. So these are the 3 basic pathways we're going to see in the sympathetic nervous system. We have preganglionic neuron to ganglia in the sympathetic chain, out to our effector neurons. So the second nerve in our chain, remember the second neuron is our postganglionic neuron. So that's after the ganglion. I'm going to jump down to the bottom. So second type of pathway would be preganglionic neuron out to a collateral ganglion, which I said we'll find them on blood vessels. There are 3 important ones that eventually, hopefully, you'll see in lab, basically on different parts of the aorta, controlling different organs in your abdomen. So then we'd have our shorter little postganglionic neuron in dark blue out to the effector organ there as well. Our third type is really specialized. So our preganglionic neuron go all the way out to the adrenal medulla, where we would find specialized cells that are kind of like neurons, kind of like adapted neurons, but we're going to call them specifically what they are, which is chromaffin cells. So those take the place of our postganglionic neurons in this case. If we wanted to look a little more closely at those pathways, this wasn't necessarily, didn't have to be part of your answer for the question I asked specifically, but it is something you should know. That the way our sympathetic nerves are working, specifically with respect to the sympathetic chain, is pretty specific. So we have our cell body of our preganglionic nerve and our lateral horn, right, of the thoracolumbar spine. Comes out the ventral root. When the ventral root, so that's on the side of the stomach, joins with our dorsal root, so that's like coming out the side of our back, but it doesn't innervate the back specifically. It would innervate like afferent nerves, so sensory stuff. When they join together, they form something called a spinal nerve. So that's what we would see coming out, those intervertebral foramina on the side of the spinal column. And our preganglionic nerve is then going to jump off the spinal nerve into the sympathetic chain. So we can see this light blue jumping into the sympathetic chain. So that's via the white ramus because it's myelinated. This preganglionic neuron is myelinated. Then we have the cell body for our postganglionic neuron in the sympathetic chain itself. That's kind of why we have ganglia. We need space for those cell bodies. So we have the cell body for our postganglionic nerve. We have a gray ramus. So the gray arm, unmyelinated, returns us to the spinal nerve. And then that postganglionic neuron continues along that spinal nerve, eventually getting where it's going to our effector organs. Okay. questions about the sympathetic nerves that we covered on Friday that may have arisen over the weekend, or just seeing this again before we move on to parasympathetics? Okay, cool. Before we do that, I just want to give you a reminder that about that exam 2 that we just took. You can set up an appointment with me anytime to go over that, or also your TAs. They have office hours on Tuesday and Thursday, and they're able to go over those with you as well. And for the study groups, you'll get an email this afternoon about how those are going to work. So those of you who asked me to assign you a group received a doodle poll last night or yesterday evening, so that I can group you based on times of availability. Those of you who want to make your own groups, start talking to your friends about who you want to be in your group. Okay. Cool. So moving into the parasympathetics. So the parasympathetic nervous system, still part of our autonomic nervous system. So it's like the pair of our sympathetics. So we're going to be going to similar effector organs. So we're going to have dual innervation. So like your heart will receive both sympathetic and parasympathetic innervation, but it's doing the opposite job. So it's the rest and digest. Sympathetic was our fight or flight. Our pathways are going to look similar to what we saw in the sympathetic nervous system, but they're going to be a little different. So we're still going to have a preganglionic neuron. We'll still have a postganglionic neuron, but we should immediately notice here that we don't have the sympathetic chain on here, right? So sympathetic chain was sympathetic ganglia. So the parasympathetics aren't involved in that system at all, really. Okay. So they're going out much closer to the effector organs where they'll synapse with their postganglionic neurons. Okay. Their origin is also different than our sympathetic neurons. So they're still coming originally from the central nervous system, but in this case, they're going to come from the head, from the brain. This central nervous system part will be the brain there. So from the cranial nerves that we saw in lab, or they'll come from the sacral region of the spinal cord. So down in your pelvis, we saw the sacrum in lab as well, right? They're going to come out of your sacrum. So they're not going to come out of those middle regions from the thoracolumbar sections. They're going to travel either all the way down from your brain or up and out from your pelvis, basically. This means that we're going to see much longer neurons. The vagus is one of the longest nerves in your body. It has to get all the way from your brain down into your abdomen. So that's quite a long nerve. So we'll expect to see longer preganglionic neurons when we're talking about parasympathetics. Our preganglionic neuron still communicates with our postganglionic neuron in a ganglion. Sometimes these will be basically right on the effector organs. So we're going to see very short postganglionic neurons. Okay. So here we see a picture of our parasympathetic nervous system. So we can see in purple, these purple regions, we have cranial nerves. And I should say, we're not going to memorize which cranial nerves they are. If you want to remember one specifically, remember the vagus. But in the future, if you take more anatomy classes, you'll probably learn that it's just some of the cranial nerves that carry parasympathetic innervation. So in future, that may be important to you. For now, just know that it's some of the cranial nerves. Okay. And then we'll see the sacral portion as well, contributing to the innervation. And here we see all the things they innervate. So you see immediately here why I am emphasizing from the cranial nerves. If you want to remember any of them at all, remember the vagus, because you can see the vagus is innervating your lungs, it's innervating your heart, it's innervating your stomach, it's innervating your spleen, it's innervating your pancreas, it's innervating your large intestine, it's innervating your small intestine. Vagus goes really far. It does a lot of stuff. You can see the sacral nerves are contributing parasympathetic innervation as well, but they're really just going to your large intestine, like to your colon, rectum, bladder, genitalia. They're really focused in that sort of pelvic region. Okay. If you wanted to know, the oculomotor nerve, the facial nerve, and the glossopharyngeal nerve are the other three that do parasympathetics. they're mainly doing parasympathetics in the head, which is why I'm saying not to concentrate on them particularly. We're not going to talk in super detail about parasympathetic reflexes in the eye, although they're pretty interesting. So we want to remember that our autonomic nerves are going to have mixed composition, which is to say some of those nervous fibers, when you touch a nerve in lab, you're touching many fibers. You're not touching one neuron specifically. You're touching many different neurons joined together. So if you were touching a nerve in lab, rolling it between your fingers, feeling how it feels kind of like thread, it's because it's several things kind of wound together, which will include efferent fibers, so motor fibers, so both your parasympathetics and your sympathetics. It'll also include afferent fibers. So your afferent fibers, with an A, not an E, are your sensory nerves. And they're transmitting information from receptors out in the body to the central nervous system. Visceral means they're kind of like on your organs here. So visceral, but we can think of them as out in your body. And that's really important for maintaining homeostasis because the afferent fibers are what tells us if we've moved away from our set point, right? We talked about error signals when we talked about homeostasis. So you need these sensory nerves to tell you if you need to make a change. So here we got a couple checkpoint questions. So we might want to remember which autonomic division is regulating the release of epinephrine from the adrenal medulla. Anybody want to answer? We haven't talked a ton about epinephrine yet, but we have seen that we're talking about the adrenal medulla specifically with respect to one thing, sympathetic or parasympathetic.
Sympathetic.
Absolutely, yeah. Autonomic division regulating epinephrine from the adrenal medulla is our sympathetic. The second question is a little more difficult, but actually related to the first one. So if we're thinking about the autonomic nervous system, producing more of a diffuse response, which here we mean more of like a general kind of body-wide response, we're going to be thinking more about our sympathetic nervous system, specifically because of this effect. So our neurotransmitters, which we can also think of sometimes as hormones, depending on the neurotransmitter we're talking about, coming out of the adrenal medulla, get into our bloodstream. And so we'll see much more generalized effects from the sympathetic nervous system. We'll see that the sympathetic nervous system, if we have a sympathetic response, is going to target a lot of different organ systems all at once. So our next topic is talking about chemical messengers and receptor types associated with them in each of these parts of the peripheral nervous system. So remember, peripheral nervous system out in the body, parasympathetic and sympathetic are part of the autonomic nervous system, which is part of the peripheral nervous system. And we'll get to other parts of our peripheral nervous system later. But we're still going to be talking about sympathetics and parasympathetics really in this section. Okay. So when we're thinking about neurotransmitters and receptors in the autonomic nervous system, we're going to be thinking mainly about two types of neurotransmitter. We're going to be thinking about acetylcholine, which will abbreviate ACH. We're going to be thinking about norepinephrine and sometimes epinephrine. So those terms are equivalent with noradrenaline and adrenaline when we're saying norepinephrine and epinephrine. Our book uses norepinephrine and epinephrine, so that's what I'm going to say. But just know for yourself, because I feel like in TV, they talk about adrenaline a lot more. Okay, so anytime we see one of these neurotransmitters, we know that the way a nerve signals, right? When we looked at synapses, we saw that we have our presynaptic nerve releasing a neurotransmitter being received by our postsynaptic nerve, right? And we talked about our ionotropic and metabotropic receptors on that postsynaptic nerve. we're still thinking about that basic sort of system here when we're thinking about autonomic neurotransmitters and receptors. Okay, so if a nerve releases acetylcholine, so if we're releasing acetylcholine as our neurotransmitter, we're going to pair that to cholinergic receptors. So look at this choline part, this C-H-O-L-I, cholinergic acetylcholine. So those go together. So if one nerve, the preganglionic nerve, for example, is releasing acetylcholine, postganglionic nerve is going to have a cholinergic receptor on it. We're going to see adrenergic receptors associated with norepinephrine and epinephrine. Adrenergic, so here we might want to remember that we could also call this noradrenaline because we see this A-D-R-E-N, right? Those are similar terms as well. So if we release norepinephrine, we're going to see adrenergic receptors. We'll see several types of each of these, but this is our basic breakdown. Okay. So here's a short list of where we're going to see each of these types of neurotransmitters. So here we're talking about what is getting released by a nerve. So anytime we see a preganglionic neuron, whether it's parasympathetic, whether it's sympathetic, we're going to be releasing acetylcholine. So that's nice and easy, actually, right? At this point, we do not need to think about whether we're sympathetic or parasympathetic. We know we're releasing acetylcholine as we get to those ganglia, which means we're going to have cholinergic receptors at that point on the postganglionic neuron receiving that acetylcholine. Acetylcholine also is involved in, or is released by, I should say, the postganglionic neurons of our parasympathetic nervous system. So if you want to associate one neurotransmitter with one branch of your autonomic nervous system, pick parasympathetic acetylcholine. That's a rule of thumb. It's not perfect, but it's easy and it'll get you most of the way there. So parasympathetic postganglionic neurons release acetylcholine, which means the effector organs for the parasympathetic nervous system. So if we have a preganglionic parasympathetic, postganglionic parasympathetic, and then we're having the heart, for example, right? Receiving that parasympathetic impulse, we're going to see cholinergic receptors on that effector organ as well, because they're receiving the acetylcholine. Our sympathetic postganglionic neurons, by contrast, are mainly going to be producing norepinephrine. So when we're thinking adrenergic receptors, we should immediately associate that in our mind. Adrenergic receptors, sympathetic nervous system, norepinephrine. The stars here is that we do have an exception, which are sweat glands, which will release acetylcholine, but this is a good rule of thumb. Sympathetic postganglionic neurons, norepinephrine. In the adrenal medulla, with those chromaffin cells, we have another little bit of a tweak here. In addition to releasing norepinephrine, they release a little bit of norepinephrine, but they are mainly going to be releasing epinephrine, 80%, right? Your adrenal glands, you've seen them on TV, right? Adrenal glands, fight or flight response, lots of systemic effects, right? From adrenaline, that's what's happening here from your adrenal glands. So this is also a sympathetic response. It's going to involve adrenergic receptors, but this is where we're going to see our more sort of diffuse effects as some of this is released into the bloodstream. Okay, and I'll pause for questions after we look at some of these pathways, because I always find seeing a picture helps it make a little more sense here. So right here, what we're looking at are three types of pathways for the sympathetic nervous system. So just sympathetic so far. So here we have our sympathetic nervous system, our preganglionic neuron to the sympathetic chain, and our postganglionic neuron out. So we refer to that preganglionic neuron as cholinergic. So we have a cholinergic neuron releasing acetylcholine as its neurotransmitter, which means that on the cell body, so on the soma of our postganglionic neuron, we would have a cholinergic receptor. We'll see that we actually have two types of cholinergic receptors that exist. We have nicotinic and muscarinic. We'll talk about the differences there in a second. But here we have our cholinergic receptor. But we would call that postganglionic neuron itself, we might refer to it as adrenergic because it releases norepinephrine. So you want to name the type of neuron by what it's releasing, the neurotransmitter it's releasing. So that postganglionic neuron is adrenergic, which means that on the effector organ will have an adrenergic receptor to receive that norepinephrine. Similarly thing happening with the adrenal medulla. We have a preganglionic neuron, cholinergic, releases acetylcholine, which means we have receptors for that acetylcholine in the chromaffin cells. So here we see some nicotinic cholinergic receptors. And they release epinephrine, also a little bit of norepinephrine. So we'll have adrenergic receptors on their effector organs. And the same thing again for our collateral ganglia. Preganglionic neurons are cholinergic, release acetylcholine. We have a nicotinic cholinergic receptor for our postganglionic neuron, which is itself adrenergic because it releases norepinephrine. Okay. Questions about what's going on here with the sympathetics releasing acetylcholine from their preganglionic neurons, releasing norepinephrine and epinephrine from their postganglionic neurons. All right, so we got a good system. We want to make sure that we remember when we're thinking about sympathetics, it's also kind of easy, as much as anything in this class is easy. Helpful to remember that sympathetics, when we're tracing that pathway, we're going to have two different types of neurotransmitter, because we'll see that when we're talking about the parasympathetics, we're just going to say the same neurotransmitter over and over again. So if we switch here to our parasympathetic pathway, our parasympathetic neuron, we have a preganglionic and a postganglionic again. Our preganglionic neuron is cholinergic, releasing acetylcholine. And our postganglionic neuron is also cholinergic. It also releases acetylcholine. So on both this postganglionic neuron and our effector organ, we'll see a cholinergic receptor. This one in the ganglion is nicotinic. This one on the effector organ we'll see is muscarinic. And this is another semi-helpful rule of thumb. Muscarinic cholinergic receptors are mainly going to be found on effector organs for the parasympathetic nervous system. You see, if I scroll back, where we were seeing cholinergic receptors in the sympathetics, we saw nicotinic, nicotinic, nicotinic over and over again. Parasympathetics, we see nicotinic here at that ganglion again, but now we're seeing muscarinic on our effector organ. I think, yeah. Our next slide now has our types of cholinergic receptors. So, well, I guess I didn't fill in here, but our nicotinic cholinergic receptors, we're going to see between the preganglionic and postganglionic neurons, okay, mainly. A nicotinic cholinergic receptor, what it does is it causes cation channels to open. So like it's going to let sodium in, put a little bit of potassium out. Overall, remember that we have a big sodium gradient. We have a big electrochemical driving force on sodium for our cells. So the result of this channel opening, the result of the nicotinic cholinergic receptor being activated, is that we're going to see depolarization in our receiving cell. So this makes sense. I'm gonna scroll back to our previous diagrams, all right? This makes sense because what we're seeing when we have a preganglionic neuron and a postganglionic neuron, if we think about an impulse traveling along, right? We have our action potential in the preganglionic neuron, we want to send that action potential to our postganglionic neuron, right? So we know that we want to depolarize this neuron because depolarization is basically what an action potential is. It's a structured, patterned depolarization, but it's always a depolarization. Okay. Muscarinic cholinergic receptors, which we mostly see on our parasympathetic effector organs, are more complicated type of receptor. They're coupled with G proteins, G protein coupled receptors. We've seen those before. And so their effect is going to depend on the target cell. So let's actually relate these two new words that we have for our cholinergic receptors to some words we've seen previously. So previously, right, we talked about ionotropic and metabotropic receptors, right? Now think back to our last unit. So which of these do you think would be a metabotropic receptor? A type of?
Muscarinic.
Yeah, the muscarinic cholinergic receptors are metabotropic. So remember that those are the ones that are coupled with G proteins. Those are the ones that take a little longer to work. Those are the ones that we could potentially have a slower but longer lasting response. If you're wondering why we have different words for these two, it's not terribly helpful for your uses, but I think it's kind of cool. It's due to how they discovered the nicotinic and muscarinic cholinergic receptors. They did an experiment using nicotine to activate those nicotinic cholinergic receptors. And then they did an experiment using muscarine, which is extracted from poisonous mushrooms, to find those muscarinic cholinergic receptors. Maybe I just think that's neat because I am a mushroom collector myself, but I know it, so I thought I'd share it with you. Okay. So our locations of the cholinergic receptors are summarized here. We've been talking about them. So we're always going to see them in our postganglionic neurons, all of the autonomics, right? So sympathetic, parasympathetic, we're going to see cholinergic receptors. Specifically, they're going to be nicotinic. So they're those ionotropic type of receptors. They're going to just let sodium in. They're going to depolarize. They're going to cause that action potential in our postganglionic neuron. Our muscarinic cholinergic receptors are going to be found in our effector organs for the parasympathetics, which makes a little bit of sense. We know that our muscarinic cholinergic receptors with the M are metabotropic, also with an M. So we want a slightly longer, slower response here. We're not focused on sending an action potential necessarily, but we are trying to elicit some type of response. We'll see that skeletal muscle, which we'll talk about later in this unit, is going to have nicotinic cholinergic receptors as well. So we'll see that as we start to talk about the somatic nervous system. Sweat glands are our special exception as they were earlier. So sweat glands will have some sympathetic innervation, but they will also have muscarinic cholinergic receptors. So that's just our exception that proves the rule. Okay, questions about the cholinergic receptors before we move on to adrenergic receptors? Cholinergic acetylcholine. All right, bit to wrap your head around, but we'll get there. Good, there's no big conceptual stuff right now. Okay. So the next thing we're going to talk about are our adrenergic receptors. So since we've just said that one of the big locations for our cholinergic receptors, specifically our muscarinic cholinergic receptors, is in the effector organs of our parasympathetics, where do we think we're going to see our adrenergic receptors mainly associated with norepinephrine? Sympathetic or parasympathetic? This whole lecture is really compare and contrast, right? So if we said we have cholinergic in the parasympathetics, where are adrenergics?
sympathetic.
Yes, thank you. All right, so our adrenergic receptors, we're going to see associated with our postganglionic neurons releasing norepinephrine in our sympathetic nervous system. So we have 5 types. We list them as alpha and beta receptors, each of which has subclasses. All of these are coupled to G proteins, so we could think of all of these as metabotropic receptors. And our big thing that we want to pay attention to when we're trying to tell apart our different types of receptors, if you're looking at a picture, you're going to see this second messenger that we've looked at very briefly in a picture before, a second messenger called cyclic AMP. We'll see that it's activated by all of our beta receptors. In one type of alpha receptor, we won't see it at all. In our second type of alpha receptor, we'll see that it's inhibited. So if you see activation of cyclic AMP, we're talking beta receptor. Okay. So our alpha adrenergic receptors are located in effector organs of the sympathetic nervous system, right? So we're going to see them in the sympathetic nervous system receiving norepinephrine, right? They're our most common type. We mostly see alpha adrenergic receptors. They're usually excitatory. So that means usually our final end result, right, would be depolarization. We'll see eventually cations moving in. Our alpha adrenergic receptors are going to have a greater affinity for norepinephrine than epinephrine, which I'm gonna make a short explainer aside here. So when we're talking about our adrenergic receptors, we're going to be comparing norepinephrine and epinephrine a lot because these receptors can actually respond to both types of neurotransmitter, either norepinephrine or epinephrine. But it's going to be easier for them to respond to one of these neurotransmitters than the other. So some of them are going to favor one type of neurotransmitter. So if we have both norepinephrine and epinephrine out circulating in the body, our alpha adrenergic receptors are going to respond to the norepinephrine first. More norepinephrine, we're going to see that this norepinephrine response. If there weren't any norepinephrine, they would still respond to epinephrine. We'll see similar kinds of comparisons as we move through the other receptor types. Okay. So our beta adrenergic receptors, all of them, are activating cyclic AMP. So that is to say we have our receptor, it receives epinephrine or norepinephrine, our G protein wanders off, splits, it activates some enzyme that's going to create cyclic AMP. We now have cyclic AMP circling around in the cell. That's our second messenger. And that's going to produce responses in the cell. So that's what happens with all of our beta adrenergic receptors. What changes for our beta adrenergic receptors is where we find them and their affinity for norepinephrine versus epinephrine. So our beta 1 adrenergic receptors are in pretty important places. We're going to see our beta 1 adrenergic receptors in cardiac muscle, so in your heart and in your kidneys. Super important, right? We're regulating blood flow. Actually, both your heart and your kidneys do partially regulate blood, although we won't get to the role of the kidneys until later. Beta 1 adrenergic receptors are usually excitatory. And they kind of don't care if there's norepinephrine or epinephrine. Beta 1 receptors respond equally to both. Beta 2 adrenergic receptors are quite different from our beta 1 adrenergic receptors. So they still have a G protein. They're still activating cyclic AMP because that's true for all of our beta adrenergic receptors. But we're going to see them in blood vessels and smooth muscle instead of in the heart and kidneys. They're usually inhibitory, so that's the effect on the effector organ, that cell. So they're still activating cyclic AMP, but when we say it's inhibitory here, we're going to say that that's going to cause like hyperpolarization, right, instead of depolarization. Finally, right, they like epinephrine more than they like norepinephrine. Our final class of adrenergic receptor is a beta-3 adrenergic receptor. This is located in adipose tissue, so in fat deposits, usually excitatory, and also equal in affinity for norepinephrine and epinephrine. So the way we're going to compare those to our previous ones, right? Beta-3, it's going to be easiest probably to confuse with our beta-1 adrenergic receptor, right? Because they're both excitatory, both equal for norepinephrine and epinephrine. So there we're going to focus in on the location of those receptors. And at the end, there's a big table comparing all the receptor types with all this data if you want a sort of consolidated location. All right, got time to take a look. Good. So here, what we're seeing is a comparison. Actually, no. What we're seeing here is an alpha receptor. Okay, so an alpha receptor here, we see it receiving either norepinephrine or epinephrine, binding into this receptor. We got our G protein dissociating. Okay, and then we see this response in the cell, whatever this response is. So how can we tell that this is an alpha-1 receptor? Does anybody know? What don't we see anywhere on this diagram? Does anybody remember what our second messenger is called? Quiet today. All right, so our second messenger was cyclic AMP, and we saw that that was associated with our alpha-2 receptors, which inhibit cyclic AMP, and all of our beta adrenergic receptors. So when we're scrolling through this image and we see cyclic AMP nowhere at all here on this image, we know that's an alpha-1 receptor, because regardless of what other type of adrenergic receptor we saw, it would have cyclic AMP somewhere in this diagram. And we know it's an adrenergic receptor rather than a cholinergic receptor because we see that we have norepinephrine or epinephrine coming in, right? If it was a cholinergic receptor, we'd see acetylcholine. So this image is a little bit difficult to orient. So what I want to highlight here for you is that on the left-hand side, it has a minus, and on the right-hand side is a positive, a plus. This is an image from your book. So what they're trying to tell you here is that on the left-hand side, we have a receptor inhibiting, and on the right-hand side, we have a receptor facilitating, right? So here we see the difference between our alpha-2 receptor and a beta receptor. So what we see the difference is that our alpha-2 receptor would inhibit the creation of cyclic AMP. So we would not do this pathway very much. We would stop making cyclic AMP if we triggered our alpha-2 receptor. But if we triggered our beta receptor, we'd make a bunch of cyclic AMP. So these two receptors here on this image would kind of fight against each other. And here is our big chart of these different types of receptors. So we've gone through this information. So you might sort of blank out this chart and try making it for yourself if you're good at studying with charts. Whew. Okay. So the reason we care about these things is that a lot of drugs target receptors, right? In order for a drug to have an effect on your body, right, it has to get into the cells, it has to do something. So different drugs will target different types of receptors and have different effects on them. So these are just some examples. You don't need to memorize any of them specifically for right now. But in the future, you might think about these receptors again as you learn about different drugs and what they do. If someone tells you that something is a muscarinic agonist, you want to know that that's going to be associated, right, with our parasympathetic nervous system, because muscarinic cholinergic receptors are in the effector organs for our parasympathetic nervous system. So that's why we have these different receptor types listed out with each of our drugs here because that's how they actually affect different types of cells. And we'll see that different cells have different receptor types in different distributions as well. So it's another way you can target specific organs, specific organ systems, because not every cell is going to just like have all 5 types of adrenergic receptors. So that's why we're differentiating those now as well, because as we start to talk about specific effector organs, we're going to want to know what types of receptors they have. Okay. So the final thing here we want to think about are our neuroeffector junctions. So when we talked about synapses before, we talked about synapses between a neuron and another neuron. But when we're finally getting out to our effector organs, when we're getting out to the body, we're trying to elicit a response. That neuron is going to have to signal to other types of tissue. Specifically, we're gonna see our neurons affecting muscle tissue a lot, or at least we're gonna talk about muscle tissue a lot. But first, we're going to talk a little bit about what the relationship between the neuron looks like on the neuron side and the effector organ. Okay, so if we're looking at a synapse between an efferent neuron and its effector organ in the autonomic nervous system, we're going to see kind of a specialized structure at the end of that postganglionic neuron. So when we were looking at neuron-neuron connections and those synapses, we were thinking about axon terminals. What we're going to see now at the end of a postganglionic neuron, these structures called varicosities. So they're analogous to axon terminals, but you'll see they have multiple swellings. So they're like kind of spaghetti fingers coming out from the end of our neuron, right? And they're lying vaguely near the effector organ, kind of on top of it. But we'll see that this isn't that sort of close, tight synaptic cleft that we saw when we were doing neuron-neuron. This is going to be kind of a diffuse response. It's going to send neurotransmitter to more of the cell, to a larger area than those really tight, close synapses. So we have these varicosities in our splitting out postganglionic neuron. You can see that we have these multiple swellings, right? 1, 2, 3, 4 down here. So these varicosities are where we're going to see our neurotransmitters. So in each varicosity, we'll have synaptic vesicles. They'll contain either acetylcholine or norepinephrine, depending on whether we're talking about parasympathetics or sympathetics, basically. They're going to work in a similar way to our synapses in the nervous system itself. But we want to remember that it's not just happening at the ends of these fingers. We have many swellings that kind of spread out everywhere. Okay. So our synaptic vesicles will be released in response to an action potential getting to the varicosities, just like when we were talking about the synapses. But we're going to get those neurotransmitters coming out at many more locations, kind of spread over our effector organ. So we'd have a similar course of events to when we were talking about synapses. We'll have our action potential arriving, right? Our deep wave of depolarization gets to the varicosity now instead of the axon terminal specifically. This opens voltage-gated calcium channels, just like in our synapses. Calcium triggers the exocytosis of the neurotransmitter. So these synaptic vesicles bind to the cell membrane, releasing our neurotransmitter. The neurotransmitter is going to be received by receptors on the effector organ. So neurotransmitter will bind to a receptor, might be a cholinergic receptor, might be an adrenergic receptor, depending on what our neuron is. We'll have a response in the effector organ, and our neurotransmitter will be degraded and diffused away from this area between the varicosity and the effector organ, and some of it will also be taken back up by that varicosity. So we also still see reuptake here. Okay. Sorry, just checking how much we have left. We're going to skip these checkpoint questions for now so that we can finish up the autonomics real quick. Most of this is a summary now of stuff that we've covered before. So we are going to regulate the autonomic function. So we want to remember here that we have both parasympathetics and sympathetics going to our organs, right? So we're always looking at a balance between sympathetics and sympathetics. So the way we regulate, whether we're in fight or flight mode or rest and digest mode, is that we like upregulate, we increase the activity of one of these two halves of the autonomic nervous system. So when we increase the parasympathetic activity, because we're at rest, we would decrease the sympathetic activity, but it's not like we have only parasympathetics going to that organ. We still have both. It's just that the parasympathetic activity would be dominant. So the converse, we could increase sympathetic activity. So have some excitation in response to some type of stressor. We would increase sympathetic activity and decrease parasympathetic activity, but we do still have both types of autonomics running to our organ systems. Okay. So an example of how these kind of go against each other, so how this would work. All right, say you stand up. Thing that happens when you stand up, all your blood actually gets pulled down by gravity, right? So your blood does get pulled towards the floor, pulled into your lower limbs, which means that the blood pressure being sensed, like up in your neck, right, or up in your thorax, it actually feels like your blood pressure is going down, right? Some people get woozy and see stars when they stand up. That's actually called orthostatic hypotension, right? It's actually a reaction to low blood pressure there. So our blood pressure goes down when you stand up. You have receptors, so visceral receptors, so afferent nerves, we're thinking, that detect this decrease in blood pressure, which means the way they signal this is that they have a lower frequency of action potentials up to the brain. So your brain knows that that means low blood pressure. This goes into an integration center, specifically for blood pressure. This is in the medulla oblongata. And so then the medulla is going to send out its response. So its response to this is going to be to increase sympathetic activity and to decrease parasympathetic activity. So as it does this, the heart starts to beat more, right? Your heart beats faster in fight or flight mode, right? So you're pumping more blood because of the increase in sympathetics. So you pump more blood out to your cardiovascular system, increasing blood pressure. So this would be a negative feedback loop to correct this sensation of low blood pressure. Okay. So this is just a comparison between our parasympathetic and sympathetic responses. Quiet, relaxed, rest and digest on the parasympathetics. Fight or flight, preparation for emergencies, stressful situations for our sympathetics, which we might not associate with two of these things, right? Or three of these things even. Mobilizing energy stores. So we're going to start getting ready to break down glucose to run away. Pupils get dilated, just a weird thing. And gastrointestinal and urinary functions get decreased because it's not helpful to pee yourself while you're running away. Okay. We refer to the balance here as tonic activity, which is just to say that both of these branches are active when you're at rest. When you're at rest, your parasympathetic nervous system is dominating, but both are active. Okay, sorry, I know I'm a couple minutes over time. That's it. You should know these brain regions. They're involved as our integration centers. We've looked at them already in lab. Okay, so that's it for me. Thank you guys for your patience.